Hi, this is the video to go along with your lab instructions for the proinsulin protein sequence comparison. This is going to take a little bit of patience and a little bit of working through some things to understand what you're seeing, but I'm going to walk you through all these things. You should have these instructions open next to you, so if you have that PDF up, that's a good thing. You notice that there's some embedded questions in here. I've taken some of those, not all of them, but I've taken some of them and I put them into a form for you to fill in your answers. So it'd be a good thing for you to have that open as well and take a look at that while you're working to see what you need to fill in on this form before you submit it to me. And that's what I'm going to look at to give you a grade. So on this set of instructions here, here's the website that we're going to go to and we're going to use. It's the San Diego Supercomputer Center, this thing called Biology Workbench. And the first thing I did was what you might do is accidentally went to that page. I'm going to go back to the instructions, get them up, and I'm going to open that in actually let's open it in a new window completely. Alright, so here's the Biology Workbench. First thing is click to enter Biology Workbench. I'm going to have you put your things into an account that I already have open, and it looks like, it already opened up for me. There's instructions on the PDF about how to get in to my account. So when you get there and you click Workbench, enter the Workbench, you're going to get this request for a name and a password. So put in Stacy L. Kaiser, all one word, and Proinsulin, with no quotation marks around it. And that'll get you to this page, the Biology Workbench. And when you're here, let me show you a little bit about the navigation. There's these buttons that allow you to go to different parts of this system. So in Session Tools, if you click on that, it'll bring up all this stuff that you can do with the sessions. So you can, if you look at this, you can see, wow, look at all these different student sessions that I've had, and some of them go back to 1999. Yeah, there's one. Cool group in 1999. If you want to, you can monkey around and go see what they did. I wouldn't advise that. I have no idea. So you're going to start a new session, and you're going to hit Run. It's kind of old school computer language here. And type in your session name here. So I'm going to type in Stacy Test 2014. So I know that this is my session I'm doing with you guys. And then click Start New Session. If your window doesn't look like this at all, click the Biology Workbench. If you click this, what it does is it gives you different ways of accessing these tools. You might have, you might get into a window that looks like this, and you don't know where all those choices are, all the buttons. Click this again, and you may get nothing still. Click it one more time. There's about 3 different views that it toggles between. I like to work in this view because it gives me a little window with all these options in it. How do you know you're in your session? This box right up here will always have the name of your session in it. If it ever says default, that means that you're not in the session that you thought you were going to be in. Now say you stop, you come back, you want to join your session again, and look at this great big long awful list that you have to search through. What you can do, this is my secret for a lot of things in life, is in your browser, go to Find, and find on your page, or Command F, if you know the keyboards. I want to find 2014. I know was one of the words that was in my session. So I can see, oh, there's 2014 there. Next one, there was a caffeine test that I did just a couple days ago. And here's Stacy Test 2014. I can click that radio button right there, go up to the top and say resume session, and it'll make sure that I'm in that session when I hit run. So there we are. So the first thing you're going to do is you're going to go out and find a protein sequence. So you need to go to Protein Tools, and you're going to do a multiple database search and hit run. So if we go back to those instructions, you've got your session started. You've hit run. You're going to put your session name here. Also, I'm having you put your session name over here so I can see which ones you did. And then click on Protein Tools. The engine, I know it's a bad, goofy, geeky name for this, but the engine multiple database search, then hit run. And it's going to say you got nothing in here right now. So here's what you do to do the search. You want to put Preproinsulin. That's the name of the protein we're going to look for, and you're going to go down to GB or Gene Bank Pri for primates. So let's click back to the window. Preproinsulin. GB Pri. I happen to know is one of the top ones there because there's a long list of them in here. And then hit search. And it's going to take a little bit. One of the key things I give you on the PDF for this is try not to use these back and forth buttons up here, because your computer is talking to a supercomputer down in San Diego, and you can get messed up on things if you try using those buttons instead. Instead, you want to hit return. That'll get you back to a window and you won't lose any data. So, we want the human gene for preproinsulin. We're going to click on that and we're going to import that sequence. Now, what does that mean that we imported that sequence? Well, you can go here and you can have it show you. Let me see if I can do that. View protein sequences, and what it does is, here's your sequence, the string of letters here. Now from the readings, if you didn't know about some of the basic cell chemistry stuff, I advised you to look at chapter 4. No, wait, it was in the Tangled Bank. There was a preview reading if you needed some. It was chapter 5. Evolution to raw materials. What you got to know, proteins are made up of amino acids. These are individual letters, amino acid codes for your amino acids. And this long string reads like like a page of music, or like you're reading a sentence in a book. The computer cuts it off here, but it starts again down here. So it's one long line of amino acids. So hit return, we go back, here's our gene, we've selected that. Now what do? We got a sequence saved in our session. So I show you, you can look at show records, it tells you a whole bunch of information about where it came from, who found it, when it was published. Then again, import sequences. Now we need to search for similar sequences. So this thing called BLAST-P is going to compare whatever sequence we give this thing to a whole big database of sequences, and it's going to return those in order of how close they match our original sequence that we entered. So you need to have your sequence checked before you hit BLAST-P and then run, or else it'll say you had searched with nothing. You didn't have anything for me to search against. So make sure you have those things. So when you first come here, you may not have that box checked. Check the box, scroll down here for BLAST-P, and hit run. We're going to accept all of the defaults on this and just scroll down and hit submit. Except for we gotta select a database. My bad. Okay, so the database I suggest that you pick, if we scroll down here, is SwissProt. So you're gonna select the SwissProt, and they've actually broken it out into different types of organisms. So what we're gonna do is take SwissProt for, let's just collect database. Well, let's just use a database for mammals. That's excluding human and rodents. So scroll down and hit submit. So we got 52 sequences that matched when it comes back. And when you scroll down to your return, you can see all of these sequences down here. They've got a database site, the database that we got them from. We have IDs on them. INS means insulin. That's the short abbreviation for the insulin sequence. You can see there's other things down here. IGF-1 is insulin-like growth factor. It looks a little bit like insulin, but it's not exactly like that, so you don't want to get down here in the insulin-like growth factors when you're comparing sequences. So stay up here where they all start with the INS here. You may scroll down here and recognize some of these. Rabbit, you can recognize probably pig, horse, bovine, if you know cow there, sheep. Here's the binomial name for all these organisms, but some of these, you know, you may not have any ideas. Ooh, Elephas maximus, that might be the elephant. So what I suggest to you is click on about 12 of these. Choose a wide group. Choose a few of them at the beginning there. Maybe we'll choose the rabbit. I don't know what AOTTR is. We'll look that one up. How about a horse, a cow, and a sheep? So you can hit show sequences, and all the ones that you selected will pop in to your box here, and they give you the common name now. So this one is from lowland gorilla, this one's from the chimpanzee, this one's from the orangutan, here's one from a night monkey, here's one from the rabbit, the horse, bovine, and the sheep. Alright, we're ready. Let's import these sequences. I ask you for only 6 sequences that you've chosen to compare, including the human. That's cool. I'm just going to run this with some extras so we can get some sense of how this runs. Alright, so notice here's our list. The human one up at the top is already checked. Let's run this. I'm going to run it with some extra animals just so you get to see what it looks like. I'm going to click on all of these. Alright, I got all those sequences selected. What are we going to do next? So, you've picked 6, including the human. List those out. Remember to check back here because I asked you to give me the 6 organisms that you're comparing to turn in to me. You're going to predict which ones are more closely related based on how you know what you know about them now. Now, what are you talking about? Predicted, closely related? Well, if they're closely related, their insulin molecules, their preproinsulin molecules should look more like each other, right? Because they've had less time to evolve differences in that molecule. So if they are more distantly related, they share a more distant common ancestor, then their preproinsulin molecules might have changed over time. The longer time you have, the more likely you are to have changed sequences, changed letters in that string of amino acids. And then briefly write down what characteristics, what do you know about the animals now? Why did you make that prediction? We've imported the sequences. Now it's time to align those sequences. So you remember those long strings of letters? The computer is going to line those letters up for us, and it's going to highlight where they're similar and where they're different. So we need to go back here. We have selected all that we want to compare, and we need to do what's called a Clustall W, multiple sequence alignment, and hit run. And again on this, I'm just going to accept all the default settings and hit submit. And it takes a little while to give you some information back. Remember, this is going down to San Diego. The computer is running some algorithms in the background to try to line these sequences up the best that they can, including in some places, as we scroll down here, some dashes. That means that the computer said, if I put in a bunch of dashes, I can get this sequence to align with everybody else much better than if this one started way back here. It wouldn't align at all. So the computer puts in these what are called gaps to increase the amount of alignment that it gets here. So, what's our output? Here's all our organisms. You can always go back up to the top here and find their common names. Here's how the computer lined up all of their sequences. It color codes things, if you take a look here. And it also puts symbols down underneath for different alignments. So if you look at where there's an asterisk, every organism has the amino acid F right there. And I'm going to post an amino acid decoding sheet to Moodle. There's single letter codes, there's triple letter codes, there's full amino acid names, so we need to be able to decode what these single letters are, which amino acids, but for right now we can see, there's a whole bunch of them here that are all the same. Here's one that's got a different color in it, SSSSSSS. Here's one with a P, and here's two with an S. So the computer says almost all of them have an S right there. Where it's green, you can see and look at for different patterns, and then where it's black, can you tell what's going on there in those cases? So take a look at your alignment pretty closely. Try to figure out what all these colors and these codes mean as far as amino acids across to everybody. And then if you keep scrolling down, there's a couple more places I want you to look on this. One of them here is the tree. Whoa, what's this? This is an unrooted, if you take a look up here, it says unrooted tree. What the computer does is it doesn't try to put in the common ancestor for everybody, because that would put a root in someplace on this. So what it does is it just relates everybody to each other without having a common ancestor in there anyway. Length of the lines. What are these? You'll have to go back up to the alignment up there and try to figure that one out. You can start to see now from evolutionary trees that we saw last week that there's some that share more closely, more recent common ancestors, and therefore they're more closely related. Look at this cluster out here. Who are all these guys? So from those that I chose, it's the gorilla, the chimp, the night monkey, and the human. They are all closely clustered out there. And here's the, oops, that's the night monkey. Who were all, oh, the orangutan is out there. So you begin to see, oh, there's some clustering based on relationships, probably closely related. What's the length of the line mean? That means something is going on up in these alignments up here, these letters. Is it increased differences, decreased differences? I want you to think about that. In the last place, when you get scrolling down here, here's a nice set of percentages, scores they call them here. They give you all your organisms up here, then give them a number, and then they do what's called pairwise comparisons. So sequence number 1 compared to sequence number 2 is 98% the same. 1 and 2 are gorilla and chimpanzee. Interesting. So as you look down here, you can see, wow, there's a lot of them that are pretty closely related. 99, number 1 and number 3. The gorilla and the orangutan are very closely related. Here's one that's at 100%. 1 and 9. 1 we can see is the gorilla. What's this long number of sequences? I can't remember what that is. So up at the top, here was our labels. Long number sequence was the human. The human and the gorilla, if we compare them, 100% identical. So use these pairwise comparisons and the tree and the alignment to answer the questions back here on the worksheet. Keeping an eye out for which ones does she want me to enter. I will post on Moodle visuals about preproinsulin and how it's made, and have you thinking about parts of it that are more important than others. So there's A, B, and C chains and leader sections of it. So I want you to interpret based on what's the important parts of the molecule, what can change. And then keep working through here. And you're going to have some more questions to answer. After you get your multiple alignments and you've looked at it, you may want to print it out at this point. So you're going to answer these questions, continue down. You want to import your alignment. So go up to the top, import your alignment. That clusters these all together with one box because the computer's already clustered them with that program that we used. And the next step is to check that alignment, and we want to do a box shade. And notice we've now entered into the alignment tools because the computer recognizes we have a bunch of sequences, they're already aligned, we want to do some more analysis with them. So you're going to hit box shade, you're going to hit run, default, 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 hit submit. And we're going to get a slightly different view of our alignments, slightly different colors. So what do these colors mean? Take a look at these colors. In this case, the computer's coloring all of them that are similar, and it doesn't worry about one or two that are different. So for instance, that one we looked at that was SSS with one P in there. Notice how it highlighted almost everybody's got an S there, and there's one that really pops out now as having a P that's different. Okay, so take a look at this box shade. You're going to think about the colors and what they mean here. If you want to, when you hit return and you're really itching for a rooted tree, the only problem, you're going to draw a rooted tree, the only problem with this is sometimes the computer puts in that root at wacky places. It may not be really great, but it just gives you a way of maybe wrapping your head around that tree, and you can see, okay, how does it align it with a root? So there's a whole bunch of organisms here that we saw in that other tree that were really closely related. See how short these branches are? They're all off on one branch all by themselves. If you take a look up here, who's most closely related in this part of the tree? How can you tell? Who's cousins to that group? Think of things like that. Okay, so that's that. I'm going to stop here. Send me questions. I'll be in email contact. Some of this kind of gets a little deep, and if it's during working hours, during the daytime, I can do a little Skyping with you, and I can help you interpret a little bit of this stuff. If you tell me what your session name is, I can get in and take a look at the session that you worked on and try to answer questions that way. And have fun. This is actually kind of a cool thing I like to play around with a little bit if I have some extra time. Yeah, I know. I'm a geek.